everyone, I'm Tanya. Welcome to Infinity Learn NEET by Sri Chaitanya. So today in this particular session, we will be discussing about cloning vectors, that is from the chapter Biotechnology Principles and Processes. Yes, it is one of the most important units when it comes to NEET examination. And here today I will be telling you about the cloning vectors in the most summarized way so that if you are going to revise it right before the examination, it's going to be really simple for you, right? So let's get started. So, when I say cloning vectors, so by the name itself, it's pretty clear that we require the vectors which will help us to clone the recombinant DNA, correct? So we require such vectors which will help us to clone the recombinant DNA because that's our ultimate motive, correct? What do we do usually? We make a recombinant DNA, and this recombinant DNA needs to be introduced into the host organism. So, introduction of the recombinant DNA into the host organism will be done with the help of a vector. So we require the vector to clear this particular path, right? And that's when we call them cloning vectors. Now, these vectors could either be plasmid or bacteriophages, etc. Now, why are we considering them as plasmid or bacteriophages? Because these are the extra chromosomal circular DNAs. And because they are extra chromosomal circular DNA, they have the ability to replicate within themselves without the requirement of any other DNA fragment, correct? Now, we will be discussing about the ideal characteristic features of a vector, correct? Ideal characteristic features of a vector, it basically means that what all qualities should be there in a vector so that it could help the recombinant DNA to get introduced into the host organism very easily, correct? So the first one has to be origin of replication, which we also call as ORI site. So by the name itself, it's pretty clear. When I say origin of replication, it means that the initiation of the replication will start from this site. If the recombinant DNA or the alien piece of DNA is linked to this site, so the replication will begin from this particular site. Along with being responsible for the initiation of replication, it also controls the copy number of the linked DNA. What is a copy number? Copy number means how many multiple copies will the linked DNA be able to produce, and that depends on the vector, because copy numbers per cell, they vary from vector to vector. One vector might produce 15 copies per cell, one vector might produce 100 copies per cell, so it varies from vector to vector. Now it depends on you that how many copies of your linked DNA do you want, and then only you will be deciding that which vector should we be linking the recombinant DNA with, correct? The next thing is the selectable marker. Selectable marker is basically that particular gene which will help to recognize between transformant and non-transformant, or which is also known as between recombinant and non-recombinant. Recombinant is that piece of DNA which has to be introduced into the host organism because it's a recombinant DNA. Non-recombinant or non-transformant is not having the desirable gene, correct? So by the name itself, it's clear, selectable marker. It will help us to select the correct piece of DNA or to identify between transformant and non-transformant or between recombinant and non-recombinant, correct? Usually, this could be various antibiotic resistance gene. It could be ampicillin, it could be tetracycline, it could be chemomycin, right? These are different antibiotic resistance gene, and they are, able to perform the function of a selectable marker when it comes to the host organism, especially the species of E. coli bacteria, right? Then, third is cloning site or the recognition site. Cloning site or the recognition site, okay? So when I talk about cloning site, if it's having just one recognition site, it reduces the complications, okay? So having a single recognition site is preferably good for the transformation process, because if there are more recognition sites, DNA will be getting complicated and the cloning of the genes will be complicated. More recognition site, more alien DNA will be linked, and then more cloning of the genes will happen. So that is why preferably one cloning site is better, because there are less complications back then, right? Apart from that, one plasmid or one vector which is very important is PBR322. Okay, the diagram is given in the NCERT, extremely important, right? Here, if I talk about the characteristic features, of course, it has the ORI site. It has ampicillin and tetracycline antibiotic resistance gene. It has the restriction sites as in second, BAM1, SAL1. These are the restriction sites which are present, okay? These are the antibiotic resistance gene. This is the ORI site. It also has an ROP site. ROP site is basically the site which, is responsible for the proteins which are involved in the recombination of the DNA. So yes, PBR322 is basically that important vector of the E. coli host organism, right? So that was it for today's session, where we have quickly summarized what exactly vectors are and how the cloning is being done. Now, before I end up this session, I want to remind you about the All India Test Series pack. Very interesting, very helpful with all the features available over here. You can get this in just rupees 2,999 if you apply the coupon code, that is YouTube50 off. Link is given in the description. Please go to the description, open the link, apply the coupon code YouTube50 off, and you can get this All India Test Series pack, right? Now, if you found this video helpful, please do not forget to like it. And if you haven't subscribed to the channel yet, do it right away and don't forget to press the bell icon. Thank you.